multidisciplinary study of human remains from the 3rd century mass grave in the Roman city of Mursa, Croatia. Abstract. A peculiar archaeological feature, a mass grave containing 7 completely preserved skeletons, was discovered during excavations in 2011 at the site of the Roman city of Mursa, modern-day Osijek, Croatia. The archaeological context and direct radiocarbon dating place the interment in the mid-3rd century CE. Bioarchaeological analysis revealed that all 7 individuals were adult males exhibiting numerous pathological lesions, including enthesopathies and injuries. Stable carbon and nitrogen isotope analysis suggested they consumed a mixed C3-C4-based vegetal diet with limited amounts of terrestrial animal protein and very little marine protein. Crucially, AncientDNA, aDNA analysis indicated that the individuals from the Mursa mass grave had a heterogeneous ancestry, and none demonstrated genetic continuity with the preceding local early Iron Age population. The presented multidisciplinary data strongly suggests that the studied individuals were Roman soldiers, victims of a catastrophic event occurring as a result of the crisis of the 3rd century, most probably the Battle of Mursa from 260 CE. Key findings from multidisciplinary analysis. 1. Bioarchaeological data, the individuals. Number and sex, the grave contained 7 individuals, all of whom were identified as adult males. Pathology and trauma, skeletons exhibited evidence of life stresses common in active populations, such as enthesopathies, disorders of the tendon or ligament attachment to bone. The presence of various injuries suggests a physically demanding or violent lifestyle. Perimortem injuries, the article notes specific perimortem injuries, such as a puncture wound on the manubrium and another on the right ilium of 2 different individuals, suggesting violent death. 2. Dietary reconstruction, stable isotopes. Dietary profile, carbon, $\delta 13, C, and nitrogen, $delta 15, N, stable isotope values were broadly uniform across the group. Consumption, the diet was predominantly C3-C4-based vegetal, typical for a population consuming a staple like millet, a C4 plant. Consumption of terrestrial animal protein was limited, and marine protein consumption was negligible. This profile is consistent with a Roman military diet in an inland province. 3. Genetic and ancestry analysis, aDNA. Genetic heterogeneity, the 4 individuals with successfully generated genome-wide data showed a high degree of genetic diversity. Distinct paternal lineages, they belong to distinct paternal haplogroups, N1A1, R1B1, I2A1B, and I1A3A1, further highlighting their varied geographic origins. Non-local origin, none of the mass grave individuals showed genetic continuity with the local Croatian Iron Age population. Their genetic profiles tended to plot closer to modern Northern and Eastern European populations, with one individual shifting towards the profile of modern-day Sicilians. This finding strongly supports the hypothesis that they were people mobilized from across the vast Roman Empire. Conclusion and historical context. The combined archaeological, anthropological, isotopic, and genomic evidence points toward a clear interpretation: the 7 men were not a local group but rather mobile, diverse adult males who died violently. The dating to the mid-3rd century CE coincides perfectly with the tumultuous period known as the crisis of the 3rd century, 235 to 284 CE, when the Roman Empire was ravaged by civil wars, invasions, and military coups. Mursa was a critical military and administrative center on the Danube-Limes border, and was the site of several decisive battles during this era. The authors conclude that the most plausible scenario is that the victims are Roman soldiers killed during a large-scale military engagement, most likely the Battle of Mursa in 260 CE, where the Emperor Gallienus defeated the usurper Ingenuus. The mass grave, devoid of customary Roman burial rituals, aligns with the emergency disposal of casualties after a catastrophic military event. Thanks for watching. Don't forget to like and subscribe to our channel.